Welcome to Microlab Mastery. Today's topic, Ubiquity of Microorganisms. In this lab, we're going to discuss the ubiquity of microorganisms. Ubiquity means that microorganisms are found everywhere or are universally distributed. Specifically, microorganisms are known to inhabit air, water, soil, plants, animals, humans, on the surface of objects, and in extreme environments. Additionally, these microorganisms can be found in free-living or symbiotic associations. Examples of microorganisms you might find in the air could be several types of bacteria, such as Streptococcus or Bacillus, fungi, such as Aspergillus, viruses like influenza, rhinovirus, or coronavirus, and yeast spores, such as Candida. In water, you may find bacteria like E. coli, viruses like norovirus, algae like diatoms, protozoa like Giardia, and fungi like Aspergillus. In the soil, we find bacteria like Streptomyces, fungi like Aspergillus, protozoa like amoebas and parameciums, viruses like bacteriophages, and algae like cyanobacteria. In plants, we find bacteria such as Pseudomonas, viruses like tobacco mosaic virus, protozoa like amoeba, yeast like Saccharomyces, and fungi like powdery mildew fungi. Microorganisms can also be found in extreme environments, including ones that are heat-loving like Thermus aquaticus, cold-loving like Psychobacter, salt-loving like Halobacterium, acid-loving, base-loving, pressure-loving, and dryness-loving. Microorganisms can also be found with animals, including various types of bacteria like E. coli or Staphylococcus, viruses like canine parvovirus, protozoa like Giardia, fungi, and archaea. In humans, we find microorganisms on our skin like Staphylococcus, in our oral cavity like Streptococcus, in our intestinal tract like Lactobacillus, in our nose like Staphylococcus, our throat Streptococcus, the vagina Lactobacillus, and the urinary tract Enterococcus. In today's lab, we're going to examine the ubiquity of microorganisms both on our cells and in the room we happen to be located in. In order to perform today's lab, you're going to need the following materials: sterile nutrient agar plates, sterile cotton swabs, sterile water, and a permanent marker. As a class, today we're going to sample and culture from several locations. We will begin by labeling our Petri plates. When you label a plate, please ensure you write the plate number, where the sample is taken from, what temperature the sample is going to be grown at, your table number, your professor's last name, and the day and time of your class. Next, please label 2 paper towels, one with room temperature and the other labeled 37 degrees Celsius. Plate number 1 is a negative control. This plate we will not intentionally expose to any microorganisms and hypothetically should remain sterile. This plate we will incubate at room temperature. Plate number 2 is also a negative control, but we're going to incubate it at 37 degrees Celsius. Plate number 3 we will open and expose to the air for 30 minutes and then incubate at room temperature. Plate number 4, a student will be taking a sample from their fingernails. In order to do so, please open a sterile cotton swab. Then dip that sterile cotton swab in sterile water. Then please proceed to thoroughly wipe underneath your fingernails using the sterile cotton swab. After this, you will then open up plate number 4 and wipe the sterile cotton swab across the surface of the agar plate. This plate will then be incubated at 37 degrees Celsius. For plate number 5, another student will take and rub their fingertips together and then proceed to touch the agar plate with their fingertips. This plate will then be incubated at 37 degrees Celsius. For plate number 6, please take a new sterile cotton swab and dip that sterile cotton swab in your sterile water. Proceed to wipe along the edge of your bench with the cotton swab. Wipe the cotton swab across the agar surface. And then incubate plate number 6 at room temperature. For plate number 7, again, get a new sterile cotton swab. Dip that sterile cotton swab into your sterile water. Wipe the cotton swab across a different side of your lab table. And wipe the cotton swab across the agar surface. This plate number 7 will then be incubated at 37 degrees Celsius. For plate number 8, please distance yourself from other students, open up the plate, and vigorously cough multiple times onto the surface. This plate will then be incubated at 37 degrees Celsius. For plates 9 through 30, you choose where to take the samples from and what temperature to incubate them at. Please feel free to be creative. At the end of this experiment, you will place the plates into either the room temperature or the 37 degree incubator. When you return during your next class session, you will then examine the plates for growth of microorganisms. When examining for microbial growth, you may be looking for several characteristics. For example, the morphology of the colony on the plate, such as its size or its shape, the margins of the colonies and the surface of the colonies, the color, the texture, and the optical properties of the colonies, and the elevation of the colonies. This video has been a collaborative effort by Dr. Brian Sage and Dr. Dheeraj Verma. Until next time, this has been Microlab Mastery.